keep it like
Okay.
less than 3.
Okay, fine.
Good morning, everyone. Welcome to our session 2 of second day. Sorry about the delay. So we had to cancel today's morning talk. Shona Chatterjee couldn't speak because he's not well. He apologized to all of you. He will visit again to NBRC in February, he said. And as you know, we'll get started with the next session. Suhita is sharing this session. Suhita, probably you know that this every talk is for 30 minutes. They have a like 20 to 25 minutes. The chaperones will help you with the timing. 10 to 5 to 10 minutes for the discussion. I will hand it over to you, Suhita.
Good morning. Welcome. Let's wish Shona speedy recovery. So we have Joby from we have Joby Joseph as our next speaker. He's from University of Hyderabad, and he's going to be talking about influence of temporal aspects of the old. Oh, come on, man. But anyway, influence of the temporal aspects of the olfactory code in classical conditioning.
Thank you, Sujita, for the introduction, and thanks for having me here to share what we have been working on recently for some time. So we mostly work with grasshoppers and honeybees. Um, and this today, while we're speaking about work, two parts. One, if I get time, and one part has been started by Bandita was a master student, then a series of master students worked on it, and some PhD students, Pranay, Deepak, and Pallavi is the one who did part of the modeling work. And the later portion, I will talk about the work by Sandhya, who was a PhD student with me. Okay, so olfactory stimuli sort of reach us in terms of concentration fluctuations and maybe sequence of orders as the insect is traversing the world, navigating for finding food, and maybe complex set of molecules which make up this order and stimuli. Now we can train so a bee to do learn this, associate this order with reward. So we can actually investigate the process of learning and the representation of the stimuli inside the brain by using this model system. So here is the bee sitting here, and here is an order port, and this is the proboscis of the bee. And the bee would be if you give sugar to the bee, then bee will extend this proboscis and drink sugar. And if you give an order before that, like order is presented here and followed by sugar, then we will associate this sugar with order, and thereafter when you give order, it will extend the proboscis. And if you do this repeatedly, you will see that around 80-90% of the bees would start responding to the order before the sugar is given by around 3 to 4 trial. Okay, so this is classical conditioning like in Pavlov's dog. So you will see the video here of what happened. And you should also listen to the sound of the muscle recordings that we do while we is extending the proboscis. So we have better control on what exactly goes on in the muscle at this point of time. Give the light, give the product of respond to the order, and they will be already learned. First trial adopter would respond. The response, the muscle sort of persists for quite some time after that. Now, what is going on in the bee's brain as this stimulus is actually traversing through it? So, it's olfactory stimulus is transduced by the receptors on the horn. Then it is transformed in various ways in the antennal lobe, like the olfactory bulb. Then it is transmitted to the higher brain centers, Kenyan cells, and lateral horn, where the Kenyan cells and downstream neurons are thought to be the responsible forming this association between the stimulus and the reward. And if you look at the various features of the representation in this pathway, there are a few temporal aspects to this representation, which is what I would talk about. One of them is the well-known rhythms in the field potential and the subthreshold of the neuron. Okay. So it is generated, rhythmic activity is generated by the antennal lobe in response to the orderant, and because these postsynaptic neurons can receive these synchronized spikes, you will see in the field potentials there as well these beta rhythms in the range of 20 hertz. Okay, so that's one aspect of the temporal code. The other aspect is that when the operand is presented for a short pulse, say 1 second or so, then you observe that these are different neurons in the antennal lobe of the honeybee, and you observe that uh, the activity of these different neurons for the same operand are different. And not only that, for the same neuron itself, if you observe over time, you observe these temporal patterns, which will consist of increase in firing rate, decrease in firing rate, and so on. Though actually the ordering stimulus itself is rather constant. It will not be like exact rectangle, but still it will not be fluctuating like this. But you would see this fluctuating representation very reliable because you can see multiple trials and all of them have similar patterns. And this then goes into the Kenyan cells, and in the Kenyan cells, you can see that these spikes are riding on the subthreshold oscillations, and this temporal pattern in turn causes some Kenyan cells to spike early, some Kenyan cells to spike later, and so on. And because of which there is a temporal aspect in the population that's responding at any given time in the Kenyan cells also. Right. So if you observe the uh, vector of responses in the antennal lobe, and it is changing over time, and if you use this vector as a representation in the 3D space as a reduced dimension, what you observe is that initially it is in this baseline state, and then it traverses with it quickly towards in around 1 second and reach this point. And and at this point, if you continue the ordering, usually it stays in this point. The representation sort of remains constant, and that's what people call fixed point in the pure order pulse. Okay, and then thereafter it will come back to the baseline. Okay? So this is the time course of the representation of vector. Okay. Now, if you look at the people usually look at it in this way, like over trials, what happens to the percentage of bees responding. But however, if you look at what happens to the muscle activity itself in these trials, what you observe is that these are two different bees. This is the first trial, second trial, third trial. You usually see that, of course, first trial there is no response, and then over trials you see an advance in the response in the muscles. So you see that the muscle response is motor response is advancing with trials as the bees are learning. So there is a temporal aspect which is missing in this representation of percentage of bees responding. So this is the code in the olfactory pathway, and this is the representation of the muscle activity over averaged over number of bees PSTH, muscle activity spike record. So what you observe, of course, first trial is responding only to the sugar, which is in this time, and this is the time where order is applied, and this is a very standard protocol used for training the bees. 4 second order, around 3 second sugar, and then what you observe is that the second trial is responding like this, and third trial, and so on. So this is the advance that I talked about in the response to the muscles. Okay. Now the circuit which is involved in this process, in this what we understand about this olfactory learning is that there is a neuron called WUM which is present in the bees, which would respond whenever sugar is presented. This is the work from Hammer, very classic old work. Uh, and muscle also would respond. But if you activate WUM, muscle will not respond. After having learned, then if you give order, you will see that the worm responds to the order. And so people think that worm is representing the reward either by directly the sugar or after having learned in response to the order which is associated with sugar. So that is one representation in the bee's brain which represents the reward in some sense. And of course, rest of the olfactory pathway is there. So these are anterior lobe, lateral horn, mushroom body, and this worm innervates all these centers. Okay, and worm expresses octopamine. If you give octopamine to these centers in substituting for worm activation, it will act as if it is reward and will associate. So which means worm activation is sufficient to cause the association, just like sugar was given. So this is the circuit sort of in this. Now, if you want to incorporate the temporal aspects of this learning within the trial, then one of the existing learning learning algorithms which can capture that and which can also have this advance which takes place with repeated trial is what is TD learning. Okay, so if you we can sort of roughly identify the variables in the TD learning in this picture. Okay, but they are not exact. So somewhere there is a V which would correspond to prediction of the reward, and there would be a delta which we don't know where it is. However, if you run the simulation with this kind of a algorithm, TD learning, and with ST, which is represented by sequence of pulses, and the reward, a square pulse, then you would expect it to have a response which would capture the muscle activity. And like this with advance, right? So, and if you do the simulation, you see clearly that there's an advance in the response with trials in the TD learning, which is not surprising. And if you run what is called extinction trials, so after B has learned this association, if you give just the order alone without reward, what you would see is that B starts forgetting, meaning lesser and lesser number of Bs would respond in later trials. And if you look at the temporal aspects of that we recorded, we are expected to find something like this in the which is what TD learning algorithm would predict. And when we make the measurement in the extinction trials, yes, it does capture that. So both this advance and the sort of receding of the learning in the by TD learning in a protocol where 4-second order pulse is used for training and testing is quite quite evident. However, what happens is that bees can also do what's called trace conditioning. So if you give a small pulse of order and then you very delayed without any overlap with the CS, you give a US like in here. Two examples are shown here, this and this with varying intervals. What you observe is that yeah, the same feature of advancement of the learning is taking place, but you also observe something, which is that this this response here and much later than here, and this peak is lower than this, and so which would mean that the temporal code that we saw for the CS representation, which is present here, which is not usually what happens here, as we saw that code keeps on changing, would actually be associating with the reward in the first trial. And then as the trials progresses, you see that earlier and earlier representations of the CS gets associated with that. If you do TD learning model with this trace conditioning, then you would see that it would not capture this, it would continue responding throughout, does not capture this feature. So we went back to the drawing board and then we said, okay, let us not bother about an existing algorithm and try and fit it, but let us look at the circuit, okay? And one additional feature that we know about the bee is that if you train the bees with an order for some trials like this and learn, and then of course it will recall for number of trials after that. You know, it can remember for many hours. However, if you take an order A and repeatedly present that order A without any reward, so you may wonder, you know, it doesn't have any meaning, meaning there's no sugar or any punishment following that. What you observe is that bees do learn something about the order, and how we know that is because you take those bees which has been exposed, you may say habituation or adaptation or so, then you try to train those bees again. Then you would see that those bees which have been habituated or exposed to the order without reward for longer number of trials would take longer time to come back up. So there is some negative reinforcement in some sense taking place without reward for the CS representation. Right? So this which would imply that not only just this WUM, which is the reward representation, there should be another representation which would also cause this negative reward somewhere. We don't know whether it's an inhibition or whether it is making the plasticity of the synapses more resistant to change when the reward comes. We don't know, but there is an inhibition to the change. So if you sort of put these variables in, trying to see what are these different representations, and with simple correlated, Hebbian type of mechanism for plasticity, where there is a positive set of weights which would be captured by the pathway from CS to the US, enhanced by the WUM, and a negative one which would be presented by as if it is a attenuation of the weights or a negative input which is learned, and plasticity mechanisms are like this, and then the total motor output which is governed by both the positive rewarded and negative rewarded component. Then you observe that we capture this fact, which is that in the So here I have represented the temporal pattern of CS by just simple 2 PNK so that we can look at it closely. And we observe that indeed with this older stimulus, 4 second and 1 second, 4 second CS, 1 second reward, you observe that indeed the second trial is here and then it shifts to earlier times, just like we observed in the experiment. And when we do the extinction trials, you can see that the extinction takes place. Now, if you do the trace conditioning, you can see that with varying delay between the CS and the US, you can see this, you can already see compared to the earlier, the third trial is little lower than the lower and earlier than the later trial. And if you increase the trace delay further, you can observe that actually the shift comes gradually. So we believe that from this, what we sort of, where am I on time? So we believe that from this, what we understand is that in the temporal code, we can learn the different components within that code, and not only that, if you have a train of either because same order produce different codes or the frequency of orders come in, which would imply different codes, then if the reward comes at some point later in the time, it can latch on and like in a what you call entire pathway from the starting of the stimulus to the end, which is consistent but maybe made up of different components, can be learned and associated. Okay. However, you should realize I still don't understand why the B responds like this, because if the reward is appearing here, you should expect the B to be smart enough to respond here. B does not do that. B all the time responds when the CS itself comes. Okay. So you should why if you look here, the other thing which you should keep in mind is that so this black is the this dirty yellow is the test trial for 1 second pulse. Okay, and you can see that it comes on and then decays off. But for a bee trained with 1 second pulse, if you give a 4 second pulse, so you have to understand, you would think that okay, these portions of the code would never be encountered by the bee, right? So if you test with 4 seconds for a bee trained with 1 second, it should not respond to these things. But you see that it is responding to a later point in time. However, what we know from the trajectory is that by 1 second it has reached the fixed point. So this is not surprising given the stimulus. Currently, we are doing experiments to check that if you give a smaller pulse here where it would make sure that the fixed point is never reached, then whether this is consistent or whether for the 4 second pulse, then it will start off decreasing at an earlier time. So another aspect, 5 minutes is including questions or excluding questions? Okay, fine. Yeah, so the other aspect, as I said, okay, fine. So the other aspect, so what we have talked about so far is that the sequence of representations which takes place, which is called the temporal patterning in the olfactory pathway, uh, has in some sense a meaning in the sense that each of those patterns can get associated, and later patterns when they associate can transfer that association to the earlier pattern. Right? So that's how the shift happens. Now, the other aspect um, of the temporal code is these oscillations that we talked about, which is generated in the antennal lobe. And so, what I told you was that in the antennal lobe, these oscillations are generated when the order is presented. However, there are some finer features of that phenomena which we should know. One is that when you present the order the first time, there are no oscillations. And if you present within tens of seconds repeatedly the same operand, which could happen in the nature, then you would see that these oscillations build up. So there is some plasticity in the AL which makes sure that this is happening. And these oscillations are generated by a recurrent excitatory inhibitory network. So there is a PN which excites an LN, then LN inhibits PN for a short while, and that time scale of that inhibition is of the order of 50 milliseconds. So that's why you get a 20 hertz oscillation in this circuit. And however, if you have built up this oscillation, now you wait for 10 minutes. What you observe is that you would this oscillation power will go away, and the next trial, first trial, would have no oscillatory power. So this plasticity has a short time of the order of 10 minutes, because of after which it will recover from that. And whether this oscillations plays a role in this learning is done was tested by Mark in this very classic paper. And the way that is was done was, you block that inhibitory circuit in the AL by applying picrotoxin, so you can show that it abolishes this oscillation. And then you train, and then what you observe is that if you train with order A and you test with order B, the B's can discriminate, provided B is an order which is very different from A. So, for example, if you train with hexanol and test with octanol, it cannot distinguish between hexanol and octanol. But if you train with hexanol and test with geraniol, then it will discriminate. It will not respond to geraniol. So the fine discrimination was compromised by removal of this oscillation. So that's what we know about this. So that people think that oscillations play a role in the olfactory coding in the sense of it is required for forming this fine discrimination. The other feature is that if you increase the concentration, you observe increased oscillation power. So these are the two phenomena which are to be kept in mind when we talk about the uh, um, uh, role of oscillations in this learning and memory, and why that is important. So in these training trials that I talked about, and in this experiment, there is there is an interval between the trial in which you give a order and then reward, and then you wait for some time before presenting the next one, which is called the ITI, inter training training interval or inter trial interval. Okay. Now, usually the most effective training trial interval is of the order of 10 minutes, which means you wait for 10 minutes before you give the next trial. And what this tells us is that, according to this observation, if you wait for 10 minutes, the first trial would look like this. It's a situation where there are no oscillations, so which is somewhat contradictory. So why should it be that there is no oscillation when actually the learning is most effective in the protocol, the 10 minutes? So that casts some doubt on the role of oscillations as a method for, in some sense, encoding information, right? So we thought we'll test that in slightly different way, and that came from the observation of, this is a work in hieroglyphus banyan. This is a grasshopper we work with. We standardized to be similar as a Schistocerca, in which most of the work is done. And this is the classical observation, right? If you give first trial and then you wait for repeatedly present, it becomes red. The black is the first trial, red is the last trial. And so what you observe is that if you don't filter this LFP, you observe not only that oscillation buildup, but there is a decrease in this deflection component. And that's not surprising, because these oscillations are created by GABA A inhibition in the AL, which naturally kills off spikes in between to create these oscillations. And this is the field potential in the calyx, which is driven proportional to the sort of spike rate coming in from the AL. So Shilpi quantified this deflection component, and what you observe is that with trial, as the oscillation power builds up, this deflection component dramatically reduces, which means that total input to the Kenyan cells are dramatically reduced over repeated presentations of order, in which interval is much less than 10 minutes. And we could see the same thing in bees. We tested whether that's true. So oscillations build up, deflection component decrease. And in the bees, the oscillations are not as reliable as uh, in the locust. So we had quite a bit of trouble to show this experiment. However, because we think this is consistent with the GABA coming in, and our view of this is that these oscillations are in some sense a byproduct of GABA plasticity trying to do gain control. So what happens when you give higher concentration, there is more power. When you repeat it present, there is more power. Both of these are conditions which are consistent with requirement of gain control. So what we are thinking is that system is trying to do gain control with GABA plasticity. However, as a consequence of that, because these systems are discrete event systems, spikes are discrete event, and discrete IPSVs are coming in, because of which these oscillations are generated. So if you simply say, oh, system is trying to do gain control, and if you do implement that in the brain, you would naturally get these oscillations. So it may be actually gain control, but not, if requirement is not necessarily those. Now, I'm not saying that this not cannot be tapped into for coding. Okay, so I'm just saying that this, if the system is just trying to do gain control, you invariably will get oscillation. And because oscillations were difficult to see in bees. So we actually captured that as this change in the deflection from first trial to second trial, which is observed here as the measure of this plasticity. And we can do the simulation of this network in the AL. And if you do that, and if you quantify the deflection in the model, you can see all the properties that you observe in terms of increasing concentration, power build up with concentration, and change in the deflection, which is measure of this oscillation, is corresponding to what you observe in the animal and consistent with gain control. So we, instead of doing picrotoxin, we varied these two parameters. One is inter-trial interval, which will control the oscillation, and also the percentage of concentration. So we have a combination of concentration and inter-trial interval, which would give a range of oscillations in your parametric space. Okay, and if you do that as expected, with concentration, the power goes up, which is the change in deflection in this case. We are not calculating oscillation, we are calculating the change in deflection from first trial to the second trial. And you will see the, as expected, the change in deflection varies with both repetition interval as well as the concentration as expected. Now, in these same intervals, we can train the bees. Now, we are not sticking to 10 minutes. We are sticking to all these intervals in which we have this concentration or the LFP measurement. And what you observe is that what you would expect is that this is the condition where which would favor maximum, which is in which we observe maximum oscillation. Okay, 2 minutes interval and 100% concentration. However, when we look at the discrimination performance in these bees, we observe that that's happening at 2 minutes and 10% interval. So we have a counter example where situation in which oscillations are not maximum, but is a condition where actual discrimination is the best. So, so we think that our view that system is trying to do gain control and oscillations are a byproduct in the olfactory system. That's the story that we are sort of pushing. So I'll stop here and take questions. And I want to also tell that this system is, I present a very simplistic picture of the system. And this is a circuit that we know of in the olfactory system in insects. Okay, so very fairly complex.
We have time for one.
Is there a change? Have you looked at like something, factors like stress or something, how it can change the nature of these oscillations?
Not really. So
But
we
you
assume
can test it potentially.
we can test it. And currently, yeah, we can.
That maybe have some understanding for any kind of social signals or how it can change. If you consider odor as some kind of a social signal, how
Yeah.
you know some
We
kind
haven't really thought about the way to look at it. Yeah, but there may be an interesting way to test. Yeah, so I don't know what really expect in terms of the change in the.
you know the something analog of attention in the bees.
Possible, we can actually do in olfactory system. I'm not sure, but in visual system it does work. It does show things like optomotor response and things like that. So maybe, and it will direct if you put a bar in a arena which we have built, it will direct the bar to align itself in a virtual arena.
Nice talk. My question is, in natural surrounding, odors are seldom presented in isolation. So if the combination of concentration, there are different set of weights for different set of odors, the same association would it would the same neural code be observed? The neurons that are specific for the conditional reward. So would the bees be able to isolate, or would some oscillations be observed for the odor that is specific to when it presented in a combination odors?
So two things. One is I don't know whether bees have a notion of a pure compound, right? So we are giving pure compounds, but if you look at the internal state of the bee, which is either at the antenna or in the antennal lobe, it is just a population of PN responding. So uh, that is order event, right? Order event is a set of PNs or ORNs getting activated, which may be either caused by pure orderants or mixture of orderants. So that we don't know whether it knows of these A and B, and when you present A and B, it can figure out whether it is A and B together. That we don't know very well. But we know that if you train A and B, it can do both, learn both A and B. In the nature, if this or so I'm not going to distinguish between pure and mixtures here. So let us say order A and order B, which may be mixtures or pure order, and if they appear in sequence. For example, the experiment we are planning to do in bees are leaving the hive and to a food source. We can train them to go there. Now suppose it encounters the order ends, so we have plumes in between. So it will actually encounter sequences of order while it is going towards the report. Now question is whether that would be learned as a sequence, which is what this would tell. And if it does learn, when you give that sequence, whether the bee would respond to the sugar as if sugar is present at the end. Yeah, I'm assuming that's what. So it has to be consistently present, so that latching will take place only if A and B consistently get presented in the same order. Thank you very much.
We have Supratim Ray from IIC, and he's going to talk about gamma ray as a tool to investigate natural processing in health and disease.
NBR. Actually, I wanted to unhide some slides. Can I quickly do that? Yes. Relevant to what he just talked about. That is unhide. Huh? How do I unhide? Just click hide first, and then some slides are not hidden. Yeah, so you can hide the slide, and then you can just press again. I see. Okay. I will just take a look at these, hide them, then now it's actually unhidden, so like they are not lost. All right. I'll just start. Yeah, 1 minute. Okay. This pointer. Yeah. Lights can be shown. Yeah. Yes. Yeah. Okay. Hello. All right. Good morning. My name is Supratim, and I'm going to talk about gamma oscillations. Please always remember, it's not gamma ray. Gamma ray is 10 to the power 22 hertz or something. It's the gamma rhythm, 40 hertz. Yeah, my name is also has a ray and there's gamma, so often it tends to become gamma ray. It's not gamma, it's gamma rhythm. Okay. So yeah, thanks for this wonderful opportunity. It's always good to be here. And next 30 minutes or so, we'll talk about gamma oscillations. Actually, I had hidden some slides, but after listening to Jyoti's talk, I actually unhid those because I wanted to show something that gels well with what he just talked about. Okay, so oscillations, lots of them in the brain. If you put EEG electrodes, for example, and close your eyes, you see that the signal goes back and forth like this at about 10 cycles per second. This is what is called the alpha wave, and it's one of many different waves that you can record from the brain signals, and they all occur at different frequencies and are associated with different types of cognitive states. We are interested in gamma waves because they are associated with a lot of cool sounding words like consciousness, attention, learning, memory, and so on. So for example, this is a monk who, when meditates, generates a level of gamma waves never reported in the neuroscience literature, huge gamma waves, according to this Hindu article. So, so that's why we are interested in understanding high level cognition, and we do that by studying gamma waves. Now, there's an important thing to note here, because I'm going to briefly talk about meditation later in this talk. I wanted to make one point very clear, which is that if you want to really know if there is an oscillatory signal component in the signal, what you really have to do is look at what is called the power spectral density. So this is a way in which you record the, you show the power of the signal that you're recording from as a function of frequency, okay, which is on the x-axis. So this is the power of the signal. There are 4 traces here, which corresponds to 3 different types of meditation practices. This is Himalaya yoga tradition, Isha Shunya, and Vipassana. And then this blue trace is the control. And this power spectral density, oscillations emerge as bumps. There are bumps in the power spectral density because there is more power at around 10 hertz, not at 5 or 20. It's at 10 hertz, so the power is more in the power spectral density at 10 hertz. Interestingly, for Vipassana meditators, it's a lot more than other meditators, but for these 2 meditators, it's the same as the control. But the point is, everyone has a bump in the alpha range. So alpha is a proper rhythm. Now, if you look at the gamma range, it's not really a bump. See that all meditators have more power in the gamma range, but it's not really a rhythm per se. It's a broadband increase over a large frequency range. So it shouldn't really be called even a rhythm. It's correct to say that there is more gamma power in meditators, but there is no bump in the PSD. So then is the gamma story all sort of, you know, not right? Well, actually it is. You do see bumps in the power spectral density in the gamma range also, but only in very special cases. And one case is to show images like these. These are what are called gratings, and when you look at these gratings, your visual cortex produces gamma waves. And these are genuine waves. So this is spontaneous activity. This is the power spectral density, no bump in the gamma range. But when you show a stimulus, the signal starts to oscillate and it produces a bump. This is what we call stimulus induced gamma. It needs to be always distinguished with the stimulus free broadband gamma, which is not really an oscillatory thing. And these are 2 very distinct things. So our lab actually focuses heavily on this stimulus induced gamma. Okay, so I'm going to show you plots like these. This is the power spectral density with a nice bump. So this is during stimulus presentation. This is when there is no stimulus, so there is no bump. If you subtract these 2 on a log scale, you get the change in power spectrum that has a nice bump. I will also show you plots like these. These are called time frequency plots, where you plot the change in power of the signal as a function of frequency on the y-axis and also as a function of time. Here time 0 is when this stimulus was shown. So it's very easy to generate gamma actually. Even in an anesthetized animal, for example, if you make their eyes open and show the stimulus, their brains will show these gamma waves. They don't have to be meditating or enlightened for us to get this. Showing a stimulus produces this gamma. In these kinds of plots, they show up as bright horizontal bands in the gamma frequency range, which is seen over the time when the stimulus is presented. At 800 milliseconds, we turn off the stimulus and gamma goes away. Okay. So then what's so cool about stimulus induced gamma? Why should we care? Now turns out, if you change the stimulus that you are showing to a human or to a monkey or to some other animal, it changes gamma in very deterministic ways. So you can control the gamma by showing the subject different types of stimuli. So here, for example, you show a small stimulus, there is weak gamma. If you show a large stimulus, there is stronger but slower gamma. The power spectral density shifts to the bump shifts to the left side at lower frequencies. You can think of the stimulus as a hammer, and the gamma is the response of the hammer. So you hit the head with a chotu hammer and there is weak gamma. You take a big hammer and hit, you get a strong gamma. It's another case where the hammer size is the same, but you are hitting it harder and harder, much like increasing the contrast that Joby was showing. And now when you hit the head harder and harder, gamma becomes faster and faster. The spectral density shifts to the right. So you can control the gamma and look at how it changes the stimulus. More interesting things, if you show color stimuli, so these are 3 recordings from 3 monkeys who were viewing different types of shades of colors. Just sitting in front of a screen and looking at different colors, these reddish hues produce intense gamma waves. Other colors don't. Well, they show much weaker waves. So the reason why it's interesting is that we think that gamma oscillations, these stimulus-induced gamma oscillations, reflect certain excitatory inhibitory interactions going on in the brain, especially with this parvalbumin positive interneurons, that produce this fast gamma. And we also think there's a slower gamma that I'm going to later talk about, which is because of the somatostatin network. And a lot of mechanisms in the brain, like gain control, adaptation, plasticity, normalization, a lot of mental disorders, they are associated with this EI balance. So gamma is a readout to study these things. So that is why we are very interested in studying these waves. Now, from a sort of more high level way of thinking of this, we can think of ways to understand behavior. So this is David Marr's very famous levels of analysis. You want to understand flight, which is a computation. You can think of the algorithm, which is like flapping of wings, and then how they're implemented. So you can think of these various levels of analysis. In neuroscience, we can analogous, in some ways think of behavior like attention, meditation, mental disorders. Then at the second level, we have certain mechanisms like gain control, adaptation, plasticity, and then other circuits that implement these mechanisms. So 3 levels you can think in neuroscience as well. Earlier people would think that gamma waves, so this is an example where attention is the behavior I want to study. These could be the mechanisms that can mediate attention, and then these could be certain circuit level implementations of these mechanisms. Earlier we would think that gamma oscillations may be playing a role, so they are somewhat part of this circuit. And then these are the slides that I unhid because like what Jovi was saying, we now think that gamma oscillations are actually just a reflection of these mechanisms. They're not really playing a role. We have, it's a very debated topic in the field. Nonetheless, it doesn't make gamma any less interesting because we think it is possible to link gamma oscillations to all these 3 levels. So in a way, if you want to make a bridge between circuits, mechanisms, and behavior, these oscillations can be mapped on to these different circuit mechanisms using modeling to different mechanisms by showing different types of stimuli and to different behavior, allowing us to make an indirect pathway and understanding circuits to mechanisms to behavior. So that's what we do try to do in the lab. This is the setup that we have. Here we have got humans from which we record EEG, but we also have monkeys in which we implant microelectrodes. These are chronically implanted microelectrodes in their visual cortex, which gives us access to spikes and a local version of EEG called local field potential, which is the activity of a few thousand neurons. We also have these subdural electrodes, ECOG, that we record from the monkey, as well as EEG from the monkey. So we have multiple scales in the monkey and EEG from humans. They all do the same thing. They all look at gratings. So in our lab, everyone looks at gratings. We changed the properties of the gratings, and we have a bunch of different projects. It's a very oscillatory centric lab. So everyone watches gratings and we look at these oscillations. In the monkey, we try to understand these mechanisms in more detail. This is in fact where most of our stuff goes on. But these are sort of nuanced questions that you can introduce normalization by having superimposed gratings and blah, blah, blah. Those are hard things to explain in a short talk. But this is where most of our energy actually goes in. We're also now studying a little bit of brain stimulation, how oscillations change when you stimulate. But this is sort of the front end of the lab. This is easier to explain. Whatever we find from our monkey recordings, the gratings are then shown to humans as they are engaged in certain tasks. And that is what I'm going to tell you guys about. So I'm going to tell you about two things. One is related to aging and disorders. This will take maybe 7-8 minutes. But I want to finish by showing you a couple of slides on some new experiments that we have done on meditation. Okay, so this is now, we are now coming on the human side, but we will take a little bit of a detour and look at one monkey project and then go up. So this all started a few years ago when there was a lot of sort of hype in the field that you can cure Alzheimer's disease by just inducing gamma waves. So in this case, you would just flicker a light and light produces an artificial gamma, and this was done in mice, and then these mice had were mice injected with some chemical that produces this Alzheimer's phenotype, and when they viewed these flashing lights, these amyloid beta plaques just reduced. This study is now, by the way, contested, so don't, it's not a, well, anyway, it's a contested study now. Nonetheless, created a lot of hype, and this is Li Hui Sai, the senior author of that paper. They showed that even sounds can do the trick and all that, and all you have to do is just look at a flickering light, and that's it. Alzheimer's will be cured. So our approach was a little more sort of little more tempered, so to speak. We were only interested in finding out that the stimulus induced gamma, these are not flickering gratings, these are static gratings. These produce the gamma that I showed you earlier. Do they change in any way as you age or if you have a mental disorder? Okay. For that, we just first wanted to find out the best possible grating that we would later show to the humans. So then in the monkey, we asked what kind of gratings can we produce a lot of gamma. So what we did, so this was the first experiment, and this was done by Vinay and Pooja. We implanted these chips in the monkey's brain, and the monkeys just view gratings, and the thing that we did differently was we showed these monkeys huge gratings. These are full screen gratings. Earlier we used to show smallish gratings. Even the large ones that I had told you were really small. Now these are full screen gratings. So because we thought that as you keep increasing the size, gamma will increase more and more, and it will be more better reflected in the EEG, which was our final aim. Likewise, Murthy in the lab would show the same things to the gratings to humans who would sit in front of the screen and view gratings. Now, what we found was quite surprising. These are monkey recordings. We are showing different gratings. This red circle is the receptive field of the neuron. It's not visible to the monkey. We see a nice gamma in the higher frequency, which is tuned to the orientation. So the horizontal grating produces a lot of gamma. Interestingly, we found another gamma. This is a second gamma. You see that? Which is tuned to another orientation. This is what we call slow gamma. And this is the traditional fast gamma. So when you show a very large grating, the brain produces 2 gamma waves. Same thing in the human as well. These are EEG recordings. In the back of the head, occipital area, you see a buildup of slow gamma and fast gamma. Interestingly, the reason why everyone else had missed it is that the slow gamma shows up in both monkey recordings and human recordings only when the grating is really large. The small gratings only produce the fast gamma, which is why earlier people hadn't seen this. Now, interestingly, and this is a somewhat involved slide. So this, what I'm showing you, is a measure of phase consistency between two electrodes. It's called coherence. Now, coherence, this red trace shows the phase consistency between two electrodes that are very close, only 400 microns apart. And then this one is about 3 millimeters apart, and these are in between. And then x-axis here is frequency, so how coherent the waves are, and this is the measure there for the two monkeys. So the bump that you see here is the slow gamma and the fast gamma. As the inter-electrode distance increases, so slow gamma coherence decreases only very little, while fast gamma decreases a lot. See that? What does this mean? This means that the fast gamma is a local signal. It's not coherent over large distances. But the slow gamma is more coherent. So slow gamma is sort of like a global gamma. It's a more spread out gamma. Fast gamma is a more local signal. Okay, and just keep this in mind because this will come in handy later on. 400 microns. This is a 4 mm by 4 mm grid. So these are neighboring electrodes, and these are sort of at the end of the electrode. These are the distance ranges in microns. Okay, so now let's get back to the human stuff. So this was a large study which took us a lot of time, and Murthy really spearheaded this one. This was part of a large scale study where different PIs were looking at different things. Somebody was recording MRI, somebody was recording PET, and so on. We were recording EEG. These were elderly subjects, more than 50 years of age, who were recruited, and we would collect their EEG, and then a detailed examination was done by trained clinicians from NIMANS, who would then classify either them as healthy or with some mental disorder. So this took us a lot of time, and then everybody would come, sit in front of a screen, just view these gratings, and then we would see how gamma oscillations change as you age or if you have a mental disorder. Okay, so we had most of the subjects, because they were recruited from the community, they were healthy. About 20 of them had early Alzheimer's or MCI. We didn't know them whether they are healthy or MCI before, we only found that out later. We also had about 50 subjects who were recorded twice. So what happens in healthy subjects as you age? So this is again a time frequency plot. These are actually young subjects, male and female. Females have more gamma. Females should feel very happy here. They have also more alpha. If it's an indication of being enlightened, then here it is, male versus female. What happens as you age? Gamma weakens even if you are healthy as you age. It also slows down a little. So, and this is a large, large numbers here. So even in healthy subjects, as you age, these stimulus-induced gamma become weak. More interestingly, if you look at the MCI subjects and find their age and gender matched healthy controls, we were very surprised to find that while the healthy controls produced gamma oscillations, the subjects who had MCI, this is preclinical Alzheimer's, mild cognitive impairment, or who had early Alzheimer's, their brains produced very little gamma. Okay, so it's sort of like a biomarker. If you were going to have a mental disorder like Alzheimer's, chances are that the gamma is going to be a little bit weak. Now, one question is, okay, we are looking at these stimulus-induced gamma. Are they really reliable? 1 year they come before they went to a temple, let's say. Next year they're coming, stuck in a traffic after having a fight with someone. So is the gamma going to be the same when we do the recording next year? So these are 20 subjects. They're all healthy. They are looking at the screen and getting gamma. Look, their signatures are quite different. Okay, some healthy people don't have gamma, some have a lot of gamma, and everybody has slightly different. So a shape like this, this band here, this is some fuzzy thing here, and so on. You can in fact identify a subject with very high accuracy just by looking at these gamma traces. Now they come next year, 1 year later, we do the same thing. What do we find? It's very similar. See this trace here, it's still there. This little thing is still there. The fuzzy gamma still remains fuzzy gamma. This is in females, 20 females. Again, the same trend. So it's a signature which is very reliable also. It varies from subject to subject, but for a given subject, it's very reliable. So it gives us promise that it could be a useful biomarker where you can record it every day, year, and if things start to go, change, then there is something wrong in the brain. Now here's another interesting thing. This is what we call functional connectivity analysis. It's similar to that coherence that I was telling you about. Now this is alpha, slow gamma and fast gamma. Middle-aged versus elderly, all healthy subjects. Here what you see here is a measure of functional connectivity measured using coherence. So this is a seed electrode, and how coherent is the brain signal from the seed electrode and other electrodes is shown by this color. And what you can notice is in the alpha range, this thing shrinks a little. It's a small shrink, but it's highly significant as you grow old. So the alpha connectivity shrinks, even though the alpha power doesn't really change. The alpha power here is about the same. Slow gamma and fast gamma, the power reduces, but the connectivity doesn't. Okay, so the power shrinks, but the connectivity remains more or less the same in the gamma range if you are healthy. But if you have the mental disorder, this is now controls versus cases. These are MCI subjects, and we have done careful power matching. You see that the connectivity falls for slow gamma if you have a mental disorder. Okay, so this is again showing nothing happens in alpha or fast gamma. So again, this shows that the stimulus-induced gamma can capture some aspects of mental disorders that we are showing. So we are very sort of, this is now something that we want to make accessible to others. So all the codes for all these different projects, raw data, etc., we have all made available. So I'll just finish a little bit by talking about this meditation. It's a very new thing. I'm showing this for the first time. In fact, we got the results only last week. So only a couple of slides, then I'll stop. So this is what we call Project Dhyan. So the idea is that we are going to study meditation over, starting from now over next few years. A typical task which has been done for decades now is to get a subject who either is an experienced meditator or a control. You ask them to open their eyes or close their eyes, and then they meditate in the middle, and we look at the brain signals. That's the standard way of doing things. The way we are doing things differently is that we also have these blocks where we will show these gratings before they meditate and after they meditate. Okay, so this is a new thing that hasn't been done. So we can study now how stimulus induced gamma is different in people who have been meditating for a long time versus not. But there's an additional twist here. We actually recorded from Brahma Kumari's meditators who are trained to meditate with open eyes. So their eyes are actually open when they are meditating. So you can actually show them gratings while they are meditating as well. So this is another block where they are meditating with open eyes, viewing gratings. So you can look at stimulus induced gamma not only before and after they meditate, but also during they meditate. So we don't have all the results right now. It's all very exciting. I'll show you one result, then I'll stop. And this to me is very sort of interesting. This is during the gamma protocol. They are not meditating here. Meditators and controls, age, gender match. We have done all sorts of matching here. Alpha, fast gamma, no change. But look at what is happening at slow gamma. Long-term meditators have much stronger. This is that global gamma I was talking about. A much stronger global gamma as compared to, this is the gamma that reduced when you have a mental disorder as well, as I showing the functional connectivity analysis showed. So this gives us promise that the stimulus induced gamma that we are studying are potentially biomarkers for mental disorders, and can also be modified and boosted by these meditative and other practices, and could be a useful indicator for studying high level cognition. So these are the people who did all the work, shown here in red. These are my funding sources, and thank you for your time.
Hi, great talk. So one question is, so most of the neuropsychiatric disorders, what we find is slowing of the gamma in the prefrontal regions. That is primarily what you see. And what you show is mostly in the occipital, and this is also stimulus induced. So I have two questions. One is there any other way to induce the stimulus induced gamma in other cortical regions? One. Second is, do you see any correlation of this stimulus induced gamma in the occipital region with the prefrontal oscillations that you see?
So very nice questions. The stimulus induced gamma, we are trying now to induce using auditory stimuli, because if you look back here, it's not a very exciting experience for the subject to put the head on a chin rest and stare at a screen. Wouldn't it be so nice if you can just put headphones and play sounds that will do the same thing? So there's a PhD student working very hard. So far we haven't succeeded. This seems to be very particular to visual cortex. Olfactory people, these bees and locusts, and those guys have very strong gamma, which is due to olfaction in the olfactory bulb and so on. Hippocampal guys, Rishi, now I'm pointing at you. So hippocampal guys see a lot of that in the hippocampus. Somatosensory people I don't see have seen it. So it's hard to induce. It's not a trivial matter. The occipital gamma, again, you have to be very careful whether you want to call it an oscillation. This goes back to the first thing that I was telling you about, that not everything is an oscillation. So often you see just a broadband increase like this. Now in the meditators, in the frontal areas, we are finding a bit of a bump even in the pre-stimulus period. So it seems that there is some oscillatory activity. But in lot of the papers, what we find is that you just compute the power and show that the bar plot looks bigger. That doesn't mean that there is an oscillation there. You really have to look at the power spectral density. Same with other frequency ranges as well. Okay, so this other gamma that you are talking about may not be an oscillation to begin with. You have to verify it by looking at the PhD, which is often not done. So the use of stimulus induced gamma, the use of a stimulus to induce gamma, and look at this stimulus induced gamma is sort of a new thing, relatively speaking. Huh. I see. No, so I'm not familiar with that literature. So we see a lot of papers where people do certain mental thing, blah, 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 and you look at a bunch of bar plots where things change. But power spectral density is often not shown, so we don't know whether it's an oscillatory behavior or just a broadband increase. But people in EEG, different people who are very creative versus who perform arts, like, and blah, blah, blah. All sorts of controls have been done. It's an easy enough experiment to do. But whether you can really tie it to these oscillatory behaviors is a different question. So to answer, I guess your question is that yes, people have been doing this for decades, but the part where we are, whether it's really an oscillation or not, and whether it's induced by a stimulus or not, those are sort of different questions which haven't been well explored. Okay, but in general, we look up, if you do a PubMed search, you'll find all sorts of trends with people doing different sort of things and seeing something in the EEG.
Hi. Very, yeah, very interesting. I had a couple of questions. Can you see? Huh. So, because this seems to be affected by the visual stimulus itself, I was wondering how sure are you that in older people, the ability to see itself might be differentially affected as you age. So, how do you deal with that? And the other very nice thing was that in the younger people, you have such clear correlations between year 1 and 2. Do you check that also in these older patients? In the patients also, do you see such a strong correlation across a year, or is there a specific trend across the year?
Yeah, very nice questions. So because we come from the monkey world, we are trained to really be careful about eye positions. Otherwise, we get grilled left, right, and center. So it's deeply ingrained in our system to really pay attention to if there are little jitters in the eye movements or microsaccades and things like that. So all these studies, not the meditation one, that one we are still doing, but in all these studies, we have controlled for the eye position. There's a little eye tracker here which monitors their eyes. We haven't done look at their visual acuity,
Right.
but but generally we ask, if you wear glasses, you can wear and then see, and we don't think that's a critical factor there. We make sure that they have to look at this dot. If they look anywhere outside of this dot over within some window, then that trial is not counted. We have also done microsaccade analysis, these little eye movements which are more in elderly people, but we have controlled for all of that and the results still hold. In the meditators, I think that maybe they have stabler gaze because they're trained, and so we have to rule all of that out. We are still doing that. So we will know in a couple of months where we stand, but we have all the data. With the meditators, we have head-free eye tracker. We have a sticker on their head so that they can meditate without their putting. How can you meditate with a chin rest, right? So we have all that data. It's a very accurate eye tracker, and the data that I have shown you, it's controlled for eye positions, so that any trial in which there's an eye movement or a blink or anything gets aborted. We haven't controlled for eye microsaccades yet, which we plan to do soon. But very nice question. What was your second question? Sorry, you had another one.
So the across year trial you showed for the young people.
Yeah, we don't have enough in the MCI category to make that test.
So I'll just ask a very quick question maybe in that case. So a great talk. So in one of the slides you showed that older individuals had attenuated functional connectivity in the alpha band. So did you account for the fact that they also tend to have slower alpha frequencies? So was that like an average measure of coherence, or was that adjusted for the subject specific alpha?
Yeah, so must be from Arpan's. Oh, that's Anak. Okay, so see, this is average alpha. Now the difference between this sort of a paradigm is that because you are showing a stimulus for 800 milliseconds and the rest state is for 800 milliseconds, the segment is only 500 milliseconds, so the frequency resolution is 2 hertz. Okay, so we have only 3 points, 8, 10 and 12. So if you have like 2-second segments, which is what you guys do, and your frequency resolution is 0.5, then you can talk about IA, PF and all that, which is what you guys do. We don't have that good a frequency resolution. So in that coarse frequency resolution between 8 and 12, the 3 points that we have, 8, 10 and 12, we see this trend. We just can't really do it very well. Okay, so that's why we haven't done it. We actually have eye closed data also for 20 seconds for these. It is not what we have analyzed. So maybe you can do that and see if there is any difference, because that way we can resolve the frequency resolution.
Thanks. Nice work. Lots to meditate on. Uh, so, uh, just wondering if you have a hypothesis about what might be going on. Let's say if you look at MAR level 2, right, mechanistically, in these meditators, what is producing this change in the gamma rhythm mechanistically in the meditators?
Okay, so so you have that's a great question, and that begs the question: what is generating this slow gamma, the one that is impaired here, and the one that is boosted there? So we know that the fast one is because of the parvalbumin positive interneurons interacting with the principal cells, and that's a local sort of interaction because they target the soma and so on. There is some evidence in rodents, it's not there in primates yet, that this might be due to the more spread out somatostatin network, or maybe the VIP network, which is sort of more global in some ways. So it's a slower rhythm, it's more global. We know that a lot of interactions that are going on require long inhibitory connections between far away sort of functional areas, which would be possible because of these long ranging interneurons. So I think that what we see here, because it only comes up when the grating is large, because it's at a lower frequency, because the coherence falls low, there's a lot of indications that this is a different neuronal network, and those long range interactions could be either boosted or affected if you have a mental disorder versus if you learn to sort of hold your thoughts together or whatever. And that is the network that is biased in meditators and affected in people who have mental disorders. So in rats, they actually optogenetically stimulated the somatostatin networks, and they found a bump at around 30 hertz or so. That's why we think rats or mouse, but I don't think it has been done in primates, so it's a guess right now.
Illuminating talk, and quick one in mental disorder cases where you're referring to, does this low gamma, you have conditions where people would have been meditating and are still having mental disorders and use that as subjects, or is there a way a monkey can be made to have a mental disorder and still measure slow gamma?
Yeah, those are so first one would require lots of subjects. So I'm actually very deeply committed to this project out of my own curiosity, and I plan to do this for, we just have now a setup where we would go to different centers. You need to have many meditators before you can find enough meditators who have a mental gay and then do this study, right? So we haven't done that. It's still a small enough sample set, so we don't know whether it can have a protective effect on these kind of meditative practices. We just require large data sets that we don't have. The first one, can we induce something in a monkey and study that? It's probably more feasible, but it's a hard experiment. Monkey research generally is quite hard. Implanting electrodes, injecting things to, it's something that I don't think I'll be able to get to. I just don't have the expertise. I think very few monkey people will actually be able to do this. See, in the monkey world, basically, you don't need that many monkeys because your N comes from the number of electrodes, not the number of monkeys. But the type of studies you're talking about, N comes from the number of monkeys. If you have to take a large cohort of monkeys, inject something in some of them and do a comparison. So practically, that's a very hard research question to do.
In slow gamma people, when you take so-called normal humans and look at if they have slow gamma less, they be a diagnostic or a predisposition?
No, so if you look at this, these are all healthy people. Some of them don't have gamma, others have massive gamma. There's huge personal variation. So that this is not a sure shot indicator of mental disorder. Over a cohort, you see differences across groups. Within an individual, there is huge variability. So I can't just say this person is more enlightened and fitter than this person. But the nice thing is that this trend doesn't change between year 0 and year 1. So everybody is born with some fingerprints and blah, 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 and same is your gamma and whatever as measured by this technique. That is reliable. If that changes, there's something to worry about. But you can't compare people because there's just huge variation in this signature from person to person. These are all healthy subjects. All 20 of them are healthy as per these very detailed tests. Some of them don't have gamma. About 80% people have one or of the two gammas, 20% don't. They're all healthy. Same with alpha. We do this alpha study in, in open day. Once a meditator came, 70-year-old guy was and then we studied alpha and didn't have any alpha. He was furious. Like, what the hell is this? You're saying alpha is all about calm and all that, and you can't even get alpha. So it's just huge variation from person to person. Huh?
That's okay.
That's true. Can't have that.
So while Rishi still walks to the stage, are there other ways to quantify better meditators, or the quality of meditators apart from gamma? And were there any other sort of cognitive differences in the guys who, I know they were healthy, but were there any cognitive? Did you do sort of a battery of tests to figure out if they were different in any way?
Yeah. So the correct way is to actually not study gamma per se, I would say. It's to do all these different types of cognitive tests. So there are lots of questionnaires that they fill. Usually they look at different facets of their personality. So I had a postdoc here, Kanishk, who actually is very fascinated in that. So there are all these 5-faceted multi something something tests. So he had a battery of tests. FFMQ, OHQ, happiness index, blah, blah, blah. We have done all of that. Rather, he has done all of, we haven't analyzed it. So I am an electrical, so I just looked at the EEG data. He has, and he's going to do all of that. We will see differences. I don't think, these things actually work. That's why people have been meditating for centuries, right? So, but various aspects of those things, whether it's certain as traits of their personality. So we have done those questionnaires, some of them at least, but I don't actually have the expertise or the data right now to talk about it. But we have done the expertise.
Thanks, Pratin. Welcome, Rishikesh, who's from IIC. Rishikesh Narayan is going to talk about plasticity manifolds.
First of all, congratulations to NBRC for your 25th year. And thank you for having me here for this symposium. It has been wonderful. And I also think, as I was mentioning during dinner yesterday, I think that it's a great idea to have NBRC as a nodal agency for this whole dialogue about brain behavior and the society, because I mean, it's an ideal location and there are so many experts around, and the kind of awareness, as we also kind of looked at in yesterday's program, among the general public of many of these things that we take for granted seems to be very, very low, and therefore it's a great idea to I mean bring up NBRC or make NBRC as the nodal agency for this broad dialogue between the society and neuroscientists. I wish you all the best for that endeavor as well. So I have entitled my talk as Plasticity Manifolds in the Brain. Now, for those of you, the idea of neural manifolds, so this is just manifolds, but in the plasticity space instead of in the neural activity space, basically. So this cube and other things will come clear as we go through this. Okay, so these are the people who actually did the work. I don't work anymore, obviously. So these are all the people who did the work. That's my lab website, and thanks to many of the funding agencies, we have been able to run the lab over the years, doing what we do basically. And for today's talk, I'm going to be focusing on part of the work by two people. One is Harshit, and other is Poonam. I'll be focusing only on this so that over to several other ideas, given that the talk is of shorter duration. Our lab in general works on these different scales. We are electrophysiologists, patch clamp electrophysiologists, and we started recording in vivo LFPs as well as from individual neurons recently. So initially we started out as a cellular neurophysiology laboratory, but we look at ion channel signaling mechanisms, and we also dabble into networks and behavior and systems level questions. Most of our questions are in rodents, mostly rats. We don't do that many mice experiments because we don't use genetic models basically. So in general, for today's, I'm going to be focusing on plasticity in the brain and the idea of what constitutes the basis for learning and memory. So the classic view of learning and memory is that it is something that is mediated by plasticity in synapses. There are changes that are happening in the presynaptic side and the postsynaptic side, and that is what mediates learning and memory. So that's the general accepted notion of what exactly happens in the brain as an animal learns whatever it learns. A more refined modern view, which is based upon several lines of experimental evidence, is that it's not just synapses that change in response to activity patterns or behavioral learning or even pathological conditions. When you zoom out of synapses and you look at other parts of the neuron or even look at other neurons that are present over there, other cell types that are present over there, it becomes very clear that just by focusing on one particular location and measuring only what is happening over there doesn't provide a broad picture of what is happening across in different regions, different parts of the neuron, different parts of the network when an animal is learning or encoding memory. It turns out that plasticity is basically ubiquitous. There are changes in synapses, there are changes in the protein molecules that are expressed in the neuron, the ion channels, the transporters, the pumps, the astrocytes, how exactly they communicate with neurons, and different cells that are present in the system. This is the hippocampus, that's the dentate, and you can see what are called as the engram cells, where the memory is stored more as a network kind of storage mechanism rather than in individual cells, individual synapses. So plasticity in today's world should be considered ubiquitous, and as I mentioned, there are several lines of evidence for this. But this poses a fundamental stability question. I don't know why all of them came together. So it poses a fundamental stability question as to if plasticity is indeed ubiquitous. Doesn't it mean that anything can change arbitrarily, giving you a scenario where you can't achieve stable learning? It turns out again, evolution is smarter than all of us put together. So ubiquitous plasticity does not mean arbitrary plasticity. As I mentioned, so just like you have different neurons that are present in the brain, individually, all of them, when you stimulate them with current injections, you can have them fire at very high frequencies, right? But that doesn't mean that when they are in the network, they are all going to be firing, covering the entire activity space. If you actually measure the activity of these neuronal populations that are present in the brain, they will fall in what's called as a low dimensional manifold. Right? So it's going to be falling within a subspace because of the connectivity constraints. Similar to that, if you look at the plasticity space, as to what all can change together when component X changes, does component Y also change, and what exactly are the constraints associated with it? You would see that even though if you look at individual components, they all can change independent of each other. When you look at it under normal learning conditions, you would see that not all of them do change together. When something goes up, something else has to go up. It cannot go down, basically. Right? So we came up with this cube. If you look at individual, pieces over here, they all look like they are taking arbitrary directions. If you do not know what the final goal, you will, and if you focus on only one component over here, then it feels like, oh, this is arbitrarily changing in arbitrary directions. But when you look at the structure and you know what final solution is, it is clear that there are certain components in this configuration that have to change together. You cannot have a scenario where this component and some other component can change together because they are not in a configuration where they can change together. And it's not just that. To achieve the final solution, let's say this is the learning goal, let's say, you don't have to follow one single route. There are several different routes from this configuration itself that you can take towards achieving this final solution. And if the configuration changes, if there is a different animal, as we saw already, there is considerable animal to animal variability, neuron to neuron variability. So if you take another configuration, again, there will be multiple routes, but the set of components that can change together over here are very different from the set of components that can change together from here. That is what we refer to as the plasticity manifolds. We put together a review article recently on this particular thing. So I'll tell you two stories. The first is with reference to the hippocampus, which is considered as a seat for learning and memory in mammals. And this is an experimental story. And the second one will be in the suprachiasmatic nucleus that is in a computational project, basically. So this is the hippocampus, and as I mentioned, a seat of learning and memory. So this is Poonam's slices. So you can see that these are very, very healthy granule cells that are present in the dentate gyrus. So this is how these cells will look like under a live cell microscope. And there is a electrode that she has over there using which she will record signals from there. So now what is she going to do with this? So when the animal is awake and behaving, and you record from these animals as they are awake and behaving, you will see that they fire in bursts, basically, right? So you will have these bursts, and they are not placed at arbitrary locations. They will be specifically patterned firing with reference to theta oscillations, right? So theta is 4 to 10 hertz. And you see that these bursts occur at the peak of these oscillations in this scenario. And you will see that the pyramidal neurons do show bursts, the granule cells do show bursts. So that's the kind of activity pattern that these neurons get under in vivo conditions. So what Poonam did was she took these cells, these are in slices. So she would patch onto a neuron and measure several different properties, input resistance, the frequency response properties, and give this theta pattern the firing activity, which will be very similar to what you see over here, and ask what kind of changes are occurring to these basic cellular properties as time progresses. So you can see that this is around a 45-minute experiment, and there will be something before and after. So these are 1-hour experiments, basically. Right? So what she observes is that there is a reduction in input resistance after theta burst firing. You can see that over here itself. There is a reduction in temporal summation, and there is a reduction in the activity, the gain of the neuron in lower frequencies, but not much in higher frequencies. However, despite this reduction in input resistance, she observed that there was an increase in the firing rate of the neurons. So this led to some kind of head scratching because if you observe a reduction in input resistance, you see that there is a lesser deflection for the same current injection, and that is what is referred to as a reduction in input resistance. But somehow there is an increase in firing rate, not a decrease that you would expect from here. So it took a while for us to figure out what was happening. After several different, this is just the that is a single experiment from one single neuron as to what exactly is happening. This is from a bunch of neurons, the control and the theta burst firing experiments. So you see that significant reduction in input resistance, and here you see the same data plotted in before after scenarios. And you also see that there is an increase in the firing rate after theta burst firing. So traditionally, if you see a reduction in input resistance, you would see a reduction in firing rate as well. So the question was, why is that the firing rate was increasing when the input resistance was decreasing, basically? So it took a while, as I said, after a series of experiments involving several pharmacological agents. Was the final conclusion that Poonam arrived at. It turned out it was not one channel that was changing in response to these activity patterns, but three of them were changing together, right? So you had the HCN channel changing, the invert rectifying potassium channel changing, and the persistent sodium channel changing, and the plasticity was not dependent upon calcium through NMDA receptors, but was mediated by calcium through gated calcium channels and IP3 receptors getting on. So, and it turned out that three of them were changing. If these two ion channel conductances increase, that would result in the reduction of input resistance. And because the persistent sodium also was increasing, you have a scenario where the firing rate was increasing. Right? So these are the behavioral relevant patterns, behavioral relevant patterns that were fed into these neurons, and this was the kind of plasticity that we observed. So there were multiple ion channels, three different ion channels to be precise, which together in changed in specific directions. All three of them increased, and that resulted in a reduced subthreshold excitability because of these two. On the other hand, gave increased suprathreshold excitability. We interpreted this from the engram cell perspective and showed that this could be a mechanism for bringing about encoding as well as homeostasis using the same kind of plasticity mechanisms. And so then the question becomes, is such conjunctive plasticity in multiple components along specific directions, is it a common rule across different neurons? Is it true that only one component pretty much never changes? So this is an old example from my prior work where theta burst pairing in hippocampal pyramidal neurons that results in synaptic plasticity, which is because of changes in AMPA receptors, and that is accompanied by localized intrinsic plasticity because of changes in A-type potassium current, global intrinsic plasticity because of enhanced HCN channel plasticity, and there is also SK channel plasticity, which all happen because of the single theta burst patterned activity that is present over here. So it's not one thing that changes, but multiple things change at the same time together because of the kind of signaling cascades that effectuate this. So then Harshit, who came into the lab, he he looked at our work about conjunctive plasticity, and he also realized that there was this the circadian rhythms, which are the rhythms which control pretty much everything. He realized that there were multiple ion channels that undergo plasticity in specific directions daily towards maintaining this excitability rhythm in the suprachiasmatic nucleus. During the day, you have higher firing rate, and during the night time, you have lower firing rate. And that is not because of changes in one single ion channels, but there were several of them, which were higher during daytime and lower during nighttime. And there was this single ion channel which was higher during nighttime and lower during daytime. So the question becomes, why are there so many channels changing at the same time? And is there only one route for a given neuron in a given location for it to undergo this kind of a transition and back up? And also whether the neuron, when it starts out from a specific configuration and goes to the night configuration and comes back, does it come back to the same configuration, or does it go to some other configuration which will give the same exact function, but through a different route, basically, right? So, and you also observe that it's not a, I mean, as simple a question as you think it is. So the answer to the question is compounded by the fact that there is a lot of heterogeneities across these neurons. If you look at the firing rate of these neurons during daytime, you see that it varies all over from say 2 hertz to 15 hertz, and you see that during the night time, you see that it varies from around 1 hertz to 6 hertz, basically. Right? So there is a lot of variability in the firing rates themselves. And if you look at individual components, so these are experimental results from the persistent sodium current and how they relate to the firing frequency. You see that even though you have high persistent sodium current, I mean, this is an inward current and therefore negative means higher current. So you see that there is pretty much no relationship between, I mean, you can see that, I mean, say the wild type, day type, day kind animals. So you will see that the neuron, sorry. So you have high firing frequency over here for the same persistent sodium, and you have a low firing frequency for the same persistent sodium. It's not a single ion channel that controls these different measurements, but it is a combination of all of them. So the question, the fundamental question that Harshit asked was, how do these suprachiasmatic nucleus neurons undergo stable firing transitions from day to night and night to day if several ion channels change? These are the ones that are higher during day and lower during night, and this is higher during the night and lower during the day. Simultaneously change in a heterogeneous neuronal population. So you have the base firing itself has brought about several combinations of this stuff. He took a very, I mean, elaborate and detailed way to address these questions. First, he generated a heterogeneous population of neurons, which constrained for 9 different measurements of these neurons, not just firing rate, RMP, resting membrane potential, and so on and so forth. Picked 20 of these, changed the ion channels in such a way that they would match with this physiologically observed outcomes over here. Only the BK goes down, the rest of them go up during the day to night transition. So this would give him night-like neurons. The night-like neurons were constrained by 9 different measurements from nighttime recordings. They also picked 26 of these again. Because of these transitions where these ones reduced and this one increased and made again search this plasticity space for going back to the daytime neurons, basically. Right? So this is one full cycle, but the difference is that we are not, I mean, hand tuning anything. This is an unbiased search of parameters. This is an unbiased search of plasticity space. This is also an unbiased search of plasticity space, basically. Right? So you do this, and he came up with 3 fundamental answers to the question that he started out with. The first answer was that so you can get day-like physiology, like, I mean, these 9 different measurements that I mentioned over here. You can get all of them satisfied by several different combinations of these ion channels and these calcium regulating mechanisms over there. Right? So you have all these different ion channels. That's the leak conductance and that's the calcium decay time constant. You can get the exact same kind of characteristic physiology of day neurons with several different combinations. You can see that they are distributed all over the place. This also kind of explains why in certain neurons you would get high NAP and certain neurons you would get low NAP for the same firing rate. And there were no cross correlations between the different ion channel properties associated with this 128 heterogeneous models, basically. Right? So that's the first question. And that is referred to as ion channel degeneracy, where several different combinations can give you the exact same functional outcomes. Right? Observation number 1. Now, observation number 2, from a given configuration, you can get to night and back to day in several different routes. So what he did was, as I said, he picked 20 neurons from here. and looked at how you can get night-like neurons using these search in the plasticity space, right? So 20 neurons, and he got 719 neurons from that, which tells you that there is not one way for you to be able to get from here to here. For the same neuron over here, you can get to a night neuron through several different combinations of plasticity, basically, right? So that's the second one, and you can also observe that we started out with certain ranges for these individual conductances, but when it underwent these two transitions and came back to where it should be physiologically, then you find that the distribution is larger compared to what we started out with, saying that it doesn't have to be the exact same configuration that gives you the same functional outcome. Get the same functional outcome using several different combinations. Second one now. And the third one, he asked if these plasticity routes, remember he was doing a stochastic search in the plasticity space with only the functional outcomes to be the constraining parameters. So he took all these plasticity, I mean the 6 different ion channels that were changing, and the ones that yielded the 719 neurons across these 20 different heterogeneous models. He looked at that, and sure enough, it was not occupying the entire plasticity space. There was a low dimensional manifold within which it lay for the day to night transitions, basically, right? So we confirmed this. This is TSNE. We confirmed this with UMAP, FATE, and PCA. And on the other hand, surprisingly, we did not see that there was any kind of plasticity manifold that was present for the night to day transitions. We confirmed this with de novo night neurons. We confirmed this with day to night to day transitions. Irrespective of how we looked at it, we did not see any manifolds over here, probably because of the number of increases versus number of decreases that were happening over there. So that's those are the 3 prominent things. You can get night neurons or day neurons through ion channel degeneracy. There is several routes by which you can transition from the night to day and day to night neurons, that is plasticity degeneracy, and there is a manifold along which these transitions occur. It's not an arbitrary plasticity that was happening. Even though all of them can increase, they don't necessarily increase in a specific manner. Right? So we also extended the ideas of plasticity manifold and the fact that you can get the same functional outcome through different routes to the pathological conditions. We, a bunch of like-minded us, wrote a review on how exactly you can get the same pathological conditions or back from there to normal excitability conditions because of changes in several different components. It's not a one-to-one mapping between the functional space and the parametric space. You can get the exact same pathological conditions with several different parametric differences in the parametric space, basically. Right? So overall, we think of it as a multi-scale plasticity manifold. There are these structured changes in several different molecular components, ion channels and other things, which are regulated by these kinases that are present over here. And these kinases are such that, say for instance, when we were looking at the hippocampal pyramidal neurons, MK2 is what was responsible for increasing AMPA receptor conductance and increasing the HCN channel conductance. So these constraints were coming from the molecular end of it, and that structured pattern kind of continues throughout, and it's not a one-to-one mapping between, say, synapses and learning or HCN channels and learning. It's more of a many-to-many relationship that is present, and therefore it is very important to respect the complexity of the brain rather than oversimplifying things to one-to-one relationship. Because yesterday also we were talking about how non-modular the brain is. So here is evidence that the neuron itself and the plasticity space of is not necessarily modular. Things change together. Things change together for a reason. So there is stability, there is encoding, there is continual learning for so many factors associated with it. So it's important to respect this complexity is what I want to end. Thank you. Correct. I mean, we have correct. So that's what we cover in this review. So we give examples of scenarios where there is no change in the excitability of single neurons, but you still get circuit level hyperexcitability. So there are, I mean, a plethora of cases like this, and we don't, I mean, think of it in this also as EI balance. We think of it as EI, which is the and intrinsic excitability, which is the neuronal property. We think of it as a EI-IE balance. So in the sense that, I mean, you can have a higher or lower level of EI balance. And if your excitation goes up, you can balance it, counterbalance it by reducing the intrinsic excitability of the neuron, right? So EI balance, I mean, we think of it as one component in the whole thing. I mean, as I said, we don't even think of it as just EI balance, right? Mm-hmm. So in stability, that's what we call as stability. Stability for good or bad reasons has different definitions, right? I mean, if you look at it at the network level, you would say, oh, yes. If you look at it at a single neuron level, you say it's firing rate homeostasis, or if you go down the molecular scale, you would say it is, I mean, calcium homeostasis. So at each and every scale, the notion of stability becomes very, very different, right? I mean, and what exactly the neuron is maintaining, what exactly is the homeostasis for, is not something which we have a clear idea of. So each one of us is throwing a dart at something, saying that, okay, I mean, it is calcium homeostasis, it is voltage homeostasis, it is firing rate homeostasis, it is the BA balance that is responsible. But it seems them seem to matter. I mean, you disrupt one of them, it disrupts pretty much the entire set of things, because there is, it's not one of those, I mean, radio-like systems where you pull off one thing and it doesn't do anything. This is an active, compensating, adaptive, nonlinear, degenerate system that continuously keeps on adapting to make sure that there is stability. And sometimes that plasticity itself becomes pathological. We don't know. We don't know. There are, I mean, as I said, there are hypotheses for this. Some of us think it is calcium. Some of us think it is firing rate. Some of us think it is voltage. Some of us think it's EI balance. I mean, some of us think that, I mean, it's the overall firing rate of the entire population of networks. We don't have a clear answer to this. I mean, Sweta also has worked, and she can add if she. Yeah, yeah, yeah. It's of course, I mean, the TTFL is what is driving it. This is just looking at it from a pure excitability perspective. This is just asking the simple question as to if the channel expression profiles of individual neurons is so heterogeneous, how exactly are they able to do these transitions without, I mean, seamlessly? And is there only one solution for all of them to go by certain levels and come back to exactly the same point or not? So that is the fundamental question that we are addressing. There is the network connectivity that we are not looking at, the synchrony between the different neurons that are present in the SCN that we are not looking at, the level interactions, the between the activity and the gene expression profiles and back. That feedback loop is not something that we are looking at here. So that would add additional constraints to the plasticity manifold that we are talking about. The plasticity manifold that we are talking about is purely from a functional perspective, asking, okay, if this is the transition that I have to undergo, can the plasticity spread throughout the entire space, or is it going to be constrained within a small manifold? That is the simple question that we are asking. Of course, there is a broader range thing that is present otherwise, which will also contribute to this plasticity manifold. But I mean, we are just saying that it's not arbitrary plasticity, and it is not that the system comes back to the same exact ion channel configuration. It can have a very different set of ion channels that are contributing to the exact same physiology that is present over there. That is all we are. I did not miss it. I did mention it. I did mention it when I was talking about it. We should definitely be talking about it. I mean, we have Sufyan, who was in the lab. He patched onto glial cells with IP3 in the pipette and showed that that results in plateau potentials in neurons, basically, which are known to induce plasticity. So we have a history in glial plasticity, not as long as Suhita, but the grand old lady of
We request all the speakers to come on stage. And others can actually ask the questions that they couldn't ask whilst the session was running.
I have the mic. I get to ask first. So yeah, I also had that question to Supratim. I like the idea, the way you presented in this Mars 3 levels, but I did not understand, or I didn't find any evidence to say that gamma is the sole sort of bump which can link these 3 levels. Why not alpha, theta, for that matter? Hippocampal guy, you said no, so theta should also be part of this linkage, right? Why is gamma the chosen one?
So first, it's not the only one. It is one. I didn't say there is no other one.
It's fine.
Now, the difference is that if you look at alpha, for example, certain behaviors will change it, and maybe the stimulus will presentation changes it, but it's harder to control its center frequency. It doesn't. Alpha is always between 8 and 12. Theta doesn't really vary the way gamma varies. The way we can manipulate stimuli to control gamma For example, as I said, increasing the size slows it down, increasing the contrast speeds it up. Then a lot of newer things, like if you have a grating and you superimpose another grating to make it a plate, it wipes out gamma. This is when you kick in the normalization, well-known normalization mechanism. So by manipulate, if you repeatedly show a stimulus, same stimulus, then gamma builds up over time. This is again a contested result in the sense we didn't find it. So by manipulating stimuli, you can actually invoke many different processes like adaptation, ways of inducing plasticity, normalization, gain control mechanisms. Those have a much bigger effect on gamma rhythm because its center frequency and its power changes in very deterministic ways as compared to these other physiological rhythm that have a fixed frequency and they are mainly associated with behavior. So yes, others may have a chance. I didn't say gamma is the only one, but the way you can control gamma with stimuli, you can't easily control any other rhythm. And these stimuli are then used to invoke different mechanisms. So this link between mechanism and gamma, just to give another example, suppose you have a grating that's drifting, a drifting grating, that will generate gamma. The more it drifts, the faster gamma gets. So there is one group in Russia that's trying to relate the slope of how changing the drift frequency changes the gamma power with a gain control mechanism. It's a complicated argument, but the point is you can potentially study gain control by changing the stimuli, and gamma tracks and is modulated much more than any other rhythm that I know of.
So
Hence the claim that it has a better chance of making these links.
no, gamma in general, I mean, that's like I guess that's a little bit of a decade back old view that the gamma is more related to this local communication neural assemblies versus these other frequencies. The slower frequencies are more across this large scale binding kind of signals. So one simple way to explain is this, just as sensory stimuli which you are using is primarily it's a sensory stimuli, so which is going to drive a local sort of communication among these areas, and you will get a change when you make minute changes in the sort of stimuli, sort of features in the feature space. You are going to see the gamma sort of as an encoding stimuli, versus if you have a more sort of cognitively laden task, though I don't like to use that word, but sort of a task where you are looking at sort of timbre in music, for example, or trying to get the sort of trying to hear Hindustani classical raga, for example. So there, the lower frequencies may play a bigger role in identifying what is going on, where you have a more complex sort of cognitive task in that sense, right? So again, I think isn't that the gamma, that your statement is a reflection of the kind of stimuli that we're observing? That is in a nutshell, that's what was my point is.
Absolutely. And but then again, getting back to the point, I think more the slower rhythms, it's easier to make a link between the behavioral level at the high end level. But once you get down to what kind of mechanisms are playing a role and what kind of circuits are driving it, those sorts of stimulus driven manipulations won't have a very sort of easy mapping with these slower rhythms that are reflecting something more complicated. This is not to diss other rhythms at all. I think they all play a role, but because you can control this gamma rhythm very accurately and precisely using stimulus manipulations, allows us access to these other sort of more low level features which may be not accessible for these more global rhythms that are reflecting more cognitively laden aspects, as you mentioned. Okay, but it's all the rhythms are there. We look at the whole spectrum, so we are finding a better way. But our paradigm is, I agree, that it's driven with the stimuli. So it is more sort of certain rhythms are more affected by it than others. That's a fair statement to me.
there a question for Joby? Is there a question for Joby? Oh, okay. So, by the way, you mentioned something about how your work would connect with Joby's talk, and I was wondering if Joby wants to elaborate on that.
No, he just was ending his talk by saying that even he thinks that the oscillations there are not playing a role, but rather are some sort of a reflection of it. A couple of decades ago, saying that oscillations don't play a role would lead to like very animated yeah. Now I think the field has mellowed, and just saying that something is a reflection is not considered that you're dissing the rhythm, because even if it's a reflection, it's a very powerful reflection. It's like saying, some people say, hey, oscillations are like exhaust fumes, they of a car. But if you can't open the car, then you have to make sense of the workings by studying the exhaust fumes and things like that. So if there is a reflection of a process, it doesn't make the rhythm anything less important. So I just wanted to say that because he pointed that out, but I'll let him now.
So the reason why so there are two views in some sense here, at least from the field which I come from, which is that the rhythms are actually used in coding. For example, people have connected the frequency of the rhythm 20 hertz to the subthreshold properties of the Kinesian cells, for example. So that's how you link the fact that this spikes occurring riding on top of this rhythm. So you are controlling the interval at which the spikes would have influence in the Kinesian cells by putting the rhythms in some ways. And so that is the how that temporal feature gets into the code. So there is a definite relationship between uh, the nature of that rhythm and how the postsynaptic neuron interprets that.
The subthreshold is because of some ion channel.
Subthreshold, yeah. So if subthreshold properties, where you can show resonance properties of the membrane, which would be the ion channel based thing. So this is the coupling. So that means the rhythms generated in the AL can be interpreted in a meaningful way in the postsynapse. That is the implication in the coding aspect, right? But as I was trying to say that um, at least in the widely used paradigms in learning, in which itself the role of rhythms have been tested under those same protocols. If you look really at the mechanism, as I said, the two aspects. One is the concentration and the build of operation, and which takes and repeatedly even in visual system if you present stimulus, oscillation build up at the gain point, gain control requirement region, so that we have recently seen. So so if you look at the intervals, the 10 minutes ITI, and you see that uh, you hardly get oscillations in any time when which the pairing is taking place. So it cannot be playing a role in actually in that particular paradigm, at least, right? So that's why we thought that that cannot be. So how do we test him? So that's why we changed the interval and we presented conditions where ITA is less than 10, so oscillations can actually build up, right? And concentration is the other factor. So both are consistent with gain control, right? Repetition, oscillations build up. Concentration high, oscillations build up. So that's why we think it's gain control. And if you simply forget everything else, I want to do gain control, and you put in GABA A type mechanism, you invariably will get oscillations, right? So that's why we think that even if the system is trying to do gain control, we'll get oscillations, and it need not be interpreted by the postsynaptic neuron. But now question is whether, yeah, is it interpreted? That I don't have an answer for at this point of time.
Connected to what he's saying, because it's another question. Sorry, just let me.
So, like you were telling, there were two parameters that you were working on, one ITI and one on concentration. Um, I was just curious to know. So, we know that different concentrations of odors actually influence the receptors itself,
like the firing of the receptors itself. One odor can influence multiple odorant receptors firing, and they in turn provide information to the anterior lobe. So when you were looking at the ITI, you were manipulating with the ITI to work with the oscillations, if I'm not wrong, work with the rhythms. The input from the orbital, from the receptors itself can influence the rhythm, right?
Correct. So the rhythms are manipulated because receptor neuron input going to the antennal lobe, interval at which they are coming in is changed. Right. So the receptor neuron input is the cause for both of them, because our order presentation, when we change interval, the intervals at which the olfactory receptor neurons will give input to the antennal lobe neurons, PNs and LNs, would change, which in turn would cause the plasticity to happen, and then so oscillations build up.
One of the takeaways that were there is the lesser concentrations actually enabled better differentiation between the odors.
The intermediate one.
The intermediate one, the 2% 10% concentrations with 2-minute ITI interval. So this may not be influenced. My my question was, it may not be directly implied with ITI itself. The rhythm pattern itself, it can be influenced by the input that it's actually getting from the receptors, and
Yeah,
they might. So I'm so sorry to tell this, but the differentiation might have occurred in the receptors itself before there was an information provided over here.
No, so without information difference in the receptors, anyway, they cannot discriminate. And we also don't know that there are multiple other processes which we have completely ignored, right? Plasticity mechanisms, which is in the mushroom body, which may be having its own time course, right? All kinds of stuff is going on. So what we can only tell is that in this paradigm, the conditions at which the best discrimination takes place is not the condition at which you see the maximum oscillation. You understand? So it's a counterexample for that. And it is consistent with the fact that it's an intermediate concentration and an interval at which it is sort of, not too fast, not too often. So that's why it is consistent with gain control in that sense. If you go higher concentration, then you need excess gain control. If you go lower concentration, then you have lesser input to the receptor neurons.
In the 10% concentration thing, so in a 2-minute interval thing, we are controlling for 2 minutes to have higher oscillations, if I'm not wrong, right?
Yeah, but the 30-second would have even higher.
Yes, sir. But I wanted to know if the nature of the oscillation itself is dependent, like the nature of oscillations itself. If the message from the receptors itself is controlling for the nature of oscillation, irrespective of the number of oscillations, they can be distinction, right?
Actually, we don't know. As far as we know, that stays somewhere around 20 hertz. But if you ask me whether it is 21 for one order and then 20 for another, I don't know actually. What we know is that there could be differences between orderings. Yeah, that's
Same lines of. I think one thing that we will have to keep in mind is we shouldn't be making broad statements as to whether oscillations are encoding or whether they are reflective of something. I mean, if you talk to the hippocampal guys, they will tell you that the theta is real. You put an electrode into the hippocampal neurons, you record from them in the intracellular voltage theta rhythms is 10 millivolts peak to peak on the average. So, and you have strong firing with reference to that, and there is a strong encoding of space in terms of the phase of firing with reference to the theta rhythm in the hippocampus. The breathing rhythm is real. There is an oscillation that is sitting over there. The ability of the entorhinal cortex to interpret the hippocampal output is real because the medial septum is governing the oscillations to both of them, basically. And therefore, this broad-based conclusions about oscillations are this, oscillations are that should be avoided. It should be taken up on a more system basis in the specific context rather than making generalized statement is what I would think is important. Otherwise, we will everything into one bin and say that this is all, I mean, all oscillations are non-encoding, all oscillations are reflective. That is where most of the problems come in. I mean, theta is real. You can put an electrode into the hippocampus, you will see a 200 microvolt signal peak to peak, and it is consistent. It is there when the animal is exploring. It's not there when the animal is resting. So it's real for, I mean, for everybody to see it, and the encoding is real. So everything is real over there. There is nothing fancy out there. The breathing rhythm, again, as said, is real. There is nothing fancy about it. Right?
So my question to Dr. Narayanan. So you showed that
Dr. Narayanan is my father.
So Rishi, if I may. So you showed that there are multiple possible sort of configurations of, say, the channel conductances that lead to the same activity patterns. So, but the existence of a manifold suggests that there's the certain subset is preferred over others. So where does this preference come from? My thinking could be that maybe one criteria that leads to the emergence of this well-defined manifold is perhaps energetic constraints. So maybe it is more expensive to tune one knob than another knob. So is that a good way of thinking about this?
It's the best way to think about it. So it's an important question. The general answer to that question is that there is indeed some kind of an error correction mechanism that is present over there, some kind of a broad-based, I mean, negative feedback loop, if you will, which Edelman, Gerald Edelman, who coined this term degeneracy, and he uses another term called as re-entry, which is what he used to define this broad negative feedback loop. So what exactly that stability criteria is, as to what exactly it is maintaining, which was the same question that came up earlier also, is something we have ideas about, but we don't know for sure as to what exactly the brain is interested in maintaining at a specific level. And it is also not, I mean, especially in encoding systems, I mean, if it is a rhythmogenic system, then it is easy to study this kind of degenerate behavior. Let's say, I mean, you are interested in a circuit which is going to generate oscillations at a specific frequency over and over again, right? But on the other hand, if you're looking at encoding systems like the hippocampus or the cortex, you have to adapt to a new stimulus that is coming inside, and therefore your goal cannot be just this feedback loop that is present over there, maintaining something at a specific level and thereby falling into this manifold. You should also have an additional constraint of learning this, adapting to this new system, new reality that is there, that the environment is presenting to you, basically. So the combination of it, that it has to encode and stably encode without losing homeostasis, that is what guides it towards this manifold, and therefore this manifold itself can change with time, is what we think it is, right?
So another sort of follow-up, if I may, a quick one, is that maybe one way to think about a lot of these neurodegenerative disorders is perhaps the inability of the system to come back to that manifold. So that would be like interpreting diseases as a breakdown of compensation, which is something that is not usually done. And maybe even Dr. Roy can comment on this. Is that some of these oscillatory signatures that we see that are different between different, say, groups, for example, the MCI and the normal group, maybe they are indicative of the system trying to get back to its original sort of the invariant configuration. And maybe the eventual progression to a disease is the inability of the system to get back to homeostasis, basically. So that could be one way of interpreting this.
One way of looking at it, yes. But I mean, as Mark Twain put it, all generalizations are false, including this one. So it's very important to not paint everything with that single brush. Say, for instance, in certain epileptic conditions, it is the plasticity that is trying to account for this new reality that goes on overdrive, and that leads to this epileptic condition. I mean, once you have status epilepticus and you have that initial insult, let's say, and that initial insult, it turns out, triggers certain plasticity mechanisms. And during the latent period that epilepsy kind of develops, which is the recurrence of seizures, there is this recurrent, I mean, over and over, there is this same plasticity mechanism being recruited, and that is what seems to be driving to this particular scenario, basically. So there could be scenarios where it is the plasticity going overdrive. I mean, you could have, it's a balance between plasticity and stability. In some cases, it tilts on one side over the other. In some other cases, it could be the compensation going RE. And I mean, I wouldn't also, I mean, confuse between degeneracy and compensation. The two are two different things, because degeneracy is much more fundamental. You take two cells which are right next to each other. They are giving you the same exact functional outcome using several different, I mean, two different sets of combinations of ion channels, basically. Right? So that is more fundamental. That is for you to really see in the cell itself. You don't require the exact same ion channel densities for you to get the exact same firing rate or exact same input resistance, or a combination of all of them put together, basically. So that is more fundamental. It doesn't have necessarily anything to do with compensation, right?
So, but if the compensatory theory of degenerative diseases is indeed true, then it puts a very different spin on how we interpret these biomarkers. Because now, say for example, a slowing of alpha is not necessarily a bad thing. It's actually a good thing.
We don't, I mean, that is something which we always keep in mind as to whether it is because of the homeostatic mechanism that were triggered by the problem itself, or is it a reflection of the problem? So these are questions which are, I mean, standard within the field. Much of it is unanswered, basically.
I have one question on the manifold and one comment on the same as well. So recently it was proposed that synaptic plasticity as Bayesian inference, where it's not
Synaptic plasticity
as Bayesian inference. So it's not encoding the new information as a new information, but it's encoding the error bars as well. So the fact that you are talking about stability in this manifold probably is leading from the fact that the underlying computation is not just about encoding the summary statistic, but the with that of the error bar. One can be like the sampling form of the computation might be related with this manifold that is observed as a stable manifold. And my question to you is, can there be a causal link drawn from manifold to manifold, not manifold to output? Like this particular configuration leads to this particular configuration from day to night and night to day. I'm talking specifically about circadian rhythm, but I'm interested in extrapolating this to neuroimaging like modality also, if it's possible.
There have, I mean, the Bayesian inference part, again, all generalizations. It could be, it may not be, depends on the system, depends upon what we are looking at, depending upon what we are encoding, how exactly the plasticity is triggered, and so on and so forth. Right. So the second part of it, with reference to manifold mapping, there have been several lines of studies associated with it. Now, not with reference to the plasticity manifold, but with reference to the relationship between the neural manifolds and the behavioral manifolds, right? I mean, what exactly are the kinds of changes that are happening in the neural manifold as the animal is learning, and how exactly does that change relate to what is happening in the behavioral manifold? Those kinds of questions have been asked, and mappings have been attempted, at least using broad dimensionality reduction and other mapping kinds of techniques that are out there. But with reference to plasticity manifolds, that has not been done, basically.
Okay.
Rishi, sorry, I should have known your paper, but I didn't hear about it. So I was wondering, so I was curious about how different it is when it changes from day to night versus night to day. I know you gave some why you constrained one way.
We really don't know the answer, but our guess is as follows. In one scenario, what was happening was there were like, of the 6 ion channels that were changing, 5 were increasing and 1 was decreasing. And in the other case, 5 were decreasing and 1 was increasing, the day to night and night to day transition. Maybe, I mean, in a scenario where the increases were required, you had the constraints. There was not, I mean, strong constraints on ion channel X changing versus ion channel Y changing. You could get the same output, for instance, by changing the persistent sodium or the leak, the sodium leak. And therefore, there were not many constraints on the system, and therefore it didn't fall on the manifold. That is our guess, basically. That's pretty much what we stated. We do not know the exact reasons as to why we don't have a manifold in one and we don't have in another.
The follow-up is that you expect for an oscillator that should be in training, let's say to light, especially the SCN, that there should also be a change across the state about how they will respond to stimuli. How do you capture that in this?
In this paper, we did not do that. We have not even looked at the networks. So this is purely from a single neuron perspective, just asking the question of what exactly happens there. If there is an, I mean, network input that is coming in.
Thinking network.
Okay. I mean, but the entrainment also recruits the GABAergic network that is present, and together all of them synchronously go up and come down. I mean, even if, let's say, if there is a single neuron, and if there is an entrainment that comes in the form of some perturbation or something from the external world, that would trigger an additional source of, I mean,
the expectation is that the pulse, the timing of the pulse would have differential responses.
It would, I mean, because of the phase response curve, when exactly the perturbation comes in. So in this, I mean, so if you looked at it, we are not looking at the entire 360 degrees, basically, because we don't have a plasticity rule here. We have tried that, but we have fallen short in when we did that, basically. So we don't have a plasticity rule. The right way to do this, I mean, this is more like a proof of principle that you have ion channel degeneracy. B, you have plasticity degeneracy. You can get the exact same day to night transition with plasticity in several different ion channels. And see, there is, at least in one particular transition, without even considering the TTFL, without even considering any of these other things, there is indeed a manifold that is forming. For addressing that question, we would need a plasticity rule, which would need some kind of a mechanism which ties to the TTFL also, and the perturbation that comes from the external world then affects the TTFL, and that drives the phase response curve, basically. That would be my, I mean, broad answer, which doesn't necessarily go into the details, but I mean, this will not be equipped to do that.
So if I understand a summary of all the talks is that, that there is no sort of one sort of story fit kind of all kind of possible observations of oscillations that we see. So it is multi-scale underpinnings, as you have suggested. So with that, so what it makes it difficult, that kind of an understanding, particularly when we talked about relationship to a particular disease. So this becomes a very non-specific sort of a marker. So can we really call oscillations a biomarker of anything? So if we can't, so studying oscillations, how would you envision studying oscillations is beneficial for solving problems of society, such as the one we discussed at length yesterday outside in the field? So in the context of neurodiversity or in the context of mental health, what is your vision in maybe in the next 10 years? Would we be there even, I mean, in terms of oscillations to solving these kind of problems? You can be as wild as possible in making. I don't know.
Not a useful biomarker. I mean, I think what Rishi alluded to is the context in which and the characteristics of the oscillation that you are looking at matters, right? So I think it can still be a valuable tool under different contexts, right?
Well, so for example, there are lots of mental disorders that have been shown to affect have an effect on gamma oscillations, schizophrenia, autism spectrum disorders, and so on, then begs the question that, hey, if something is a little different, uh, how much are you really figuring out about them, what kind of disorder it is? Um, and the same holds for other type of oscillations as well. So the point is well taken that it will have limited effect. It's certainly indicative of something, because it changes and could be reflecting something. I think, again, I'm biased towards gamma oscillations. The fact that it uh, it can be changed by presenting the stimulus gives us a lot more power than other rhythms. Just to give a very specific example, as I told you that if you take a grating and drift it, the faster more faster it drifts, the faster the rhythm gets, and its amplitude drops after a while. So now instead of just saying that, okay, I have an oscillation in healthy people versus control, and the power is less or more of the frequency, it's like a 1-bit information. You actually have a curve where you have several points as a function of drift rate and power for controls and for other people having some disorders. So people have shown that this fall off of how power falls with drift rate is different for autism spectral disorder and healthy controls. Similarly, for another disorder whose name I'm forgetting, but basically, the point to say is that once you can manipulate a rhythm, you can find different trends, different way with which the rhythm changes as the stimulus parameter changes, and that may depend on the mental disorder that you have. So it gives you more power because you're not just looking at an endogenous rhythm in two populations, but how a rhythm is affected by a battery of stimulus. And this is a largely unexplored field right now, which is why I think gamma oscillations are very powerful, because you can actually manipulate it in so many different ways. And then different disorders may change those trends in different ways. It's for us to then leverage this power and try to see how far we can go.
Maybe I'll quickly respond. So let me be the devil's advocate. I say in the frontal areas, meditation increases gain control. Then you would naturally see if the GABA A is involved in this gain control, you may see gamma rhythms going up. So definitely it can be signature of increased gain control, among other things, right? So that way, as a signature, I don't see what's the problem in using it. You know what I'm saying? So I don't think just saying that,
Let's take the discussion to lunch. We have to come back. Let's thank all the speakers and also the audience for asking the questions, and also to Suhita for chairing the session. We come back here at 2.